you.
Pessoal, só para vocês entenderem como é que vai se dar a nossa conversa hoje, a gente vai falar em inglês e depois, para quem não fala inglês, a gente vai dar uma versão no YouTube que vai ter tradução, tá bom? Então, espero que vocês gostem. Vamos lá. It is an honor to welcome Professor Craig Melo, American molecular biologist, professor at the UMass Medical School, and recipient of the 2006 Nobel Prize in Physiology or Medicine. Together with Andrew Fire for the discovery of RNA interference, a regulatory mechanism that can be leveraged to treat diseases. This groundbreaking discovery transformed modern biology by revealing a fundamental mechanism of gene regulation, with direct impact on the development of new genetic therapies and our understanding of epigenetic inheritance. Dr. Mello's work has paved the way for innovative medicines already in use and has inspired an entire generation of scientists. Thank you for being here, Professor.
Great to be here. Thank you, Daniel.
Thank you. We have prepared a few questions for you, and we also received a couple questions sent by our audience, including young students, professors, the Minister of Science and Technology of Brazil, and journalists, that I will be playing them. There are short clips as we move along. To begin, for our audience, many of whom are not scientists, could you please explain in simple words what you discovered and why it was considered so important that it earned the Nobel Prize? And of course, how did it feel to win the Nobel Prize?
Well, first of all, I wish I could speak in Portuguese to this audience, but I hope that they will be able to understand me. If not, I'm willing to try to go slow so that you can translate.
Perfect.
Would
Yep.
you would you prefer that, or would you prefer I just
So
go?
I would suggest we just move along, and if people have any inquiries, they can send it on the chat.
Okay, great. So, RNA interference was a real surprise for many of us. You have to remember we first began seeing these really strange responses to injected genetic information back in the late 90s. At that time, we barely had an internet. You could hardly, there was hardly any email,
iPhone, no iPhone.
no iPhone. There was no Google even,
Oh,
right? Back in 1994. Well, anyway, by 1998 or so, when we published the paper, it was just humans, society was just beginning to use guided search. Before that, we always, when we looked for information, we always had to dig out an encyclopedia and flip through this alphabetic,
yeah.
search process that was, page turning. But living things have been living in the information age for billions of years, and they are really good at handling information. And so when we naively were injecting information into this little animal, it was responding to that information. And what the organism was doing was taking chunks of the information that we were giving it and loading those as little search queries, little pieces of the genetic information were getting loaded onto a genetic search engine, a protein that is guided by genetic information to find genetic targets and then to regulate them. It would be like if you could type something into your web browser or Google on your computer, and then you could find that piece of information, but not only could you find it, you could turn it off.
Yes.
Organisms had already invented this remarkable, highly specific guided search mechanism that could find and regulate genetic information. And it had been uh, operating on this planet in every, form of life on the planet. There's guided search in bacteria, as all of you know. It's called CRISPR, and there's a whole variety of different CRISPR mechanisms. Guided search is fundamental to life because living things are they thrive on information. They need information to make the living state work.
Yeah.
And And believe it or not, the search engine that carries out this guided search inside of all of our cells in our whole body, it's doing it right now, and it's doing it at the rate, it's searching at the rate of 100 million collisions per second. It can find and regulate anything in your body, any gene in your genome. It can find and regulate the transcripts, turn them off, and and it's it's just a fundamental natural mechanism that we can now program as with medicine.
That's human beings thought they invented Google. So
Yeah.
having a Google that has millions of years, likely.
Yeah. I wish we had patented that little lowercase I after RNA I.
That's
Uh,
true.
we should have.
Every
Um,
every company is using design now, that's great. That's really, really fascinating. And Professor Melo, can you tell us a bit about how this particular discovery happened? Was the RNAi mechanism discovery planned since the beginning, or it was more like a surprise that just, happened and you said at some point, "Oh my God, we discovered something big here."
more of the latter, really. Obviously, we weren't prepared. We didn't expect that this type of response would exist. Um, when we started to see it, we almost still couldn't believe it. It was so remarkable. RNA, when genetic information, most people think about DNA, but RNA is in many ways a much more exciting molecule. It's an informational molecule that can do chemistry. It has the ability to do catalytic uh, to mediate catalysis, to actually, at the heart of the ribosome, the machine that puts together amino acids by reading the genetic code, that whole apparatus has at its heart an RNA catalytic mechanism. So RNA and is really it's like the software that's running in the cell. RNA is what makes you live. It really is the spark inside of the living cell. So the RNA uh, now maybe we should have anticipated this, but we absolutely were surprised because RNA is known to be very unstable. So when we injected it into the animal, we were, of course, really surprised that it could mediate interference that not only was incredibly robust, but also long lasting. And the silencing could spread throughout the tissues of the animal, which was really surprising because if you inject DNA into these tiny little animals that we work on, or pretty much into any animal, it doesn't move from cell to cell easily. Even DNA, which is much more stable, but the RNA was moving systemically. And so obviously there were active mechanisms at work that had to be underlying this really surprising genetic response that we named RNA interference. In 1998, when we described the phenomenon in that Nature paper that was cited by the Nobel Committee, we had no idea how it worked.
Oh, really?
Zero.
Wow.
Zero. We were speculating, of course, but we tried to keep it to an absolute minimum in the paper because we really just didn't know how it worked. But
Review. Sorry to interrupt, Professor. Just wanted to say that reviewer two didn't do a good job there. Just kidding, just kidding.
yeah, no, zero mechanistic insight, but we
That's
still we still got it published in Nature. But yeah, it wasn't until the following year that I felt we had a eureka moment, which was when we cloned the first gene involved in the mechanism. So even as we were submitting the paper to Nature, we had already initiated a genetic screen to search for mutants that knock out the pathway.
Yep.
And that genetic screen uncovered a gene we named RNAi deficient number one. That's how we often name genes. It's kind of backwards because, of course, the gene encodes the function, but when you mutate the function, RNAi no longer works. So we called this gene RDE1. And when we clone the gene, we discovered that the gene has homologs in plants, animals, and humans. And this was a very exciting moment because we now knew that this mechanism was likely shared across all domains of life.
That's absolutely fantastic. For me, it's really incredible that you study an organism that is quoting your words of the size of a comma in a printed piece of paper, and then you can extrapolate that mechanism in human beings. It's really fascinating and incredible.
Yeah, this tiny little worm that you're referring to, we shared over 3 billion years of common ancestry with this little animal.
Wow.
Uh, and the mechanisms at work are so ancient that they were already very old in the common ancestor of worms and humans back below the Cambrian, 100 million, not 100 million at least below the Cambrian. The Cambrian is this weird event in the history of life where all of a sudden animals started to get big and there's this huge blossoming of large-bodied life. But we now know that living things were present and really diverse on the planet well before that time. But I wanted to just tell you something relating to the discovery of RDE1, this gene that I referred to earlier, that was truly, truly exciting. Right. So we identified a bunch of mutants that knocked out the RNA interference response. So we could inject double-stranded RNA and the animals would not respond to it anymore. They had no response anymore. So we knew that the genes were important for RNA interference. But interestingly, some of them, like RDE1, had no other phenotype. There just was no RNAi response. But others, when you knock them out, the little transposons, these selfish elements that are present in the genomes, they make up half of the human genome, right? These little elements that can self-replicate, they're like little viruses that are jumping around in our genomes. Those began to jump. The
Wow.
worm has them too, but they began to jump around spontaneously, causing new mutations in the animal. So the pathway that we had discovered was related to the mechanism that controls the transposons in the genome of the worm and now we know of the human as well. But there was a parallel mechanism of some kind that did not control the transposons that involved this RDE1 gene. So the complexity here began already at this early stage, as early as 1999. We knew that there was RNA interference, but that there was a lot more going on. RNA interference and then some other related pathway that controls the transposons. And then of course as we were exploring these avenues to try to understand that complexity, we discovered that there's a whole pathway that is functioning with these other small RNAs called microRNAs that are so important developmentally and in all other in many other ways. And then of course in the germline, the immortal lineage where the genome is passed from one generation to the next, there was the pyrene pathway that is the pathway regulating the transposon. So the guided search mechanism that we discovered was just one of many guided search mechanisms. And the common ancestor of worms and humans already had the microRNA Argonaut system and the pyrene Argonaut system. And so there's still so much that we need to learn about how these systems work. And so it's really exciting times still in this area of research.
That's absolutely fascinating to me. Yeah, really. Um, one of the questions we wanted to ask you, Professor, relates to your personal motivation, and that relates to what first inspired you to become a scientist at all, and was there a moment or experience in your life that set you on this path, or was it like something that it always you, you were driven by?
Well, that that's an interesting question. It has a very long answer. So I would say I'll break it down into a few different parts. First of all, I was incredibly fortunate to be the child of parents who wanted their children to just pursue their passion. My mom is an artist, and my dad was a scientist, is now retired, but he's still around, and he was a paleontologist. So he worked at the Smithsonian Museum. I knew I wanted to be a scientist from very young. Um, and my desire to pursue science was cultivated by not only my parents, but my grandparents. And my grandparents on on both sides, they never had any of the chances I had. They didn't even get to finish high school. Not a single grandparent finished high school, but they were smart people, just as smart as me, hardworking, and they loved knowledge. They respected knowledge. They wanted their children to have a chance to go to college. So those grandparents of mine worked really hard jobs and made sure their kids got to go to school. So it's a really long answer, but I have to tell you, I really appreciated that opportunity. I loved the fact that I would get to just do whatever I wanted in terms of schooling as far as college. And then, so as I was approaching the graduating year or so of high school, I was sure I wanted to be a scientist, but I wasn't sure which type of scientist I wanted to be. I'm glad we didn't disappear. Uh,
Yeah.
but I read in the newspaper when I was, in high school one morning. I remember looking at this newspaper article about how they had cloned the gene encoding insulin, the protein that many diabetic patients can no longer make. And they had put the gene from the human into bacteria. And bacteria could read the human genetic code and make the human protein. I remember reading that article and thinking to myself, that is so amazing. The genetic code is the same in
Yeah.
bacteria and humans. Why is that? And I looked through that whole article, and not once did the article even mention that that was surprising.
Wow.
So for me, the whole idea that you could have an article in the newspaper and not mention something at so amazing, it just
Absolutely.
told me I got to work on this because, these adults, they have no clue. This stuff is cool. Genetics really is incredible. It's a universal language of all living things. So when I was in high school, I thought, for the first time, biology really caught my eye. It really caught my interest at that point because genetics, this new form of molecular genetics, was now an opportunity that you could explore more scientifically, in the laboratory. You could clone and you could even sequence genetic information, determine the genetic code, and compare genetic information. Anyway, that's what really got me going.
That's really nice and inspiring. Professor, perhaps um, less, complex question, but it might be complex, who knows, is a question that someone asked in the chat. How does it feel to win the Nobel Prize? Did it change something on your daily activities? And how was the moment you were let know that you won the prize?
Yeah, the prize is interesting in the way that the way that I have come to look at the prize is it is a it confers a responsibility. The prize is for the discovery, it's not for the person, you know. And Andy and I know we both feel the same way. We were incredibly fortunate to be able to be, really on the ground floor of a discovery that involved, hundreds and hundreds of scientists all around the world working in different organisms, et cetera. So the awarding of the prize is actually a very humbling experience and also comes with a new sense of responsibility, right? I mean, now you're a spokesperson for that technology. You have to help make sure that it is used wisely and safely, and of course that it does the maximum benefit for mankind. So that whole element is there, and of course the phone call is always a big surprise, usually a big surprise. They're very good at surprising people at the Nobel headquarters. That's their specialty. I know Andy and I were both 45 at the time, and we did not expect it because we thought the microRNA prize, which was awarded last year by the medicine committee, would be awarded before ours because we knew the two pathways were related to each other already by 2006. So, but nevertheless, the phone was ringing at around 4 a.m. On the first Monday in October, and
Wow.
I heard the phone, and when I walked back into the bedroom, I'd been my daughter has type 1 diabetes, and she was just a baby at the time, and she's doing fine, but this is in a poignant way related to our earlier conversation, she requires human insulin made for her by bacteria to survive. Like many type 1 diabetics, she can have a fairly normal life, but she has to constantly monitor her blood sugar and give herself insulin. I was checking her blood sugar as I often did at night, and I heard the phone ringing. And when I came back into the room, it was ringing again. My wife said, "Don't pick it up. It's a crank call." And I
It's
hope we didn't crash, did we?
no, no,
Oh,
no. We're back. We're back.
we're good. So anyway, she said it's a crank call. Don't pick it up. And I said, well, they are announcing the Nobel Prize tonight. Maybe I
Oh,
should take it.
yeah. Who knows?
So I picked it up, and it was a very nice gentleman with a very convincing Swedish accent, Hans Jornvall. And we had a great conversation. And what an amazing, tiring, long day that was. But it was super wonderful. It was really great to get to share those moments with my mom and dad, who also got to attend the ceremony, and of course, all my family and my friends and my colleagues. It was just such a wonderful opportunity to celebrate the science, and the Swedish people really do a great service to mankind in the way they celebrate scientific discovery over there. It's just truly for them, it's like, the World Cup or the Super Bowl.
Absolutely.
Yeah.
Uh, Professor, that's really a so inspiring story. Uh, I don't have enough words to really say how much you are inspiring right now, and also you will inspire with your words the new generation to come. Especially, we're going to have a recording of our conversation, so this will hopefully continue to impact lives of Brazilians, and we are speaking in English, so anyone who speaks English. Speaking of science, recently we saw the FDA approval of a drug that I believe you have vast knowledge, inclisiran, therapy-based RNA interference type of therapy. Um, and I wanted to ask you, how does it feel to see treatments being developed um, shortly after your kind of discovery, right? And in a way that it was so fast, the discovery to how to leverage that discovery in a therapeutic application, right? So could you comment a little bit on that?
Yeah, it's incredible, to know. that the therapies that are based on this discovery are already saving lives, really making a difference in the clinic. It did take quite a long time by comparison to some other Nobel discoveries, but nevertheless, 20 years or so isn't that long, that's about what it took. Um, and the first approved drug in 2018, nothing worked until the chemistry was worked out. Because delivering these molecules into a patient or into a cell from the outside is really difficult. These RNA molecules really are relatively unstable. And in a big animal like humans, injected RNA gets degraded very rapidly prior to being able to reach the target tissues and give therapy. So really, I have to say that it's the work of the chemists that really made this into a drug, and that hard work began, really in earnest right after the first evidence that this pathway could be triggered in human cells, and beautiful work from a whole host of other labs showed that as you put chemical modifications into the molecule, you could stabilize it dramatically. So you mentioned the drug inclisiran. One of the things that's so amazing about the inclisiran clinical trial is that they were able to show that these stabilized guide RNAs that can enter the search engine of the cell, these stabilized RNAs could be injected once or twice per year and affect therapy continuously for 6 months at a time in a human. So, a relatively small subcutaneous dose of this chemically stabilized guide proved not only to be incredibly safe in like 14,000 people who were injected. The only negative reactions were a little bit of redness around the site of injection. And the target gene, which uh, which causes your cholesterol levels to be high, was downregulated for months and months at a time after a single dose. So the therapy really demonstrated. I mean, compare that to insulin, for example. My
Yeah.
daughter needs to give herself insulin continuously. She's actually pumping it into her body all the time because insulin is, not only very complex in terms of its pharmacology and how it works, but also a relatively short-lived drug. Like most drugs, you have to take the pill every day. Well, RNAi is different.
Yeah.
RNAi can program this search for months at a time. And with these very stabilized guide RNAs, we can achieve long-lasting and durable regulation. Now, what that means is that it opens up the opportunity to give a drug that modulates gene expression in beneficial ways for months at a time and can confer life-saving therapy for
Yes.
patients who have no other options because there are many genetic disorders in humans that are untreatable by conventional small molecule or antibody type strategies. But if you can specifically target the underlying genetic pathway that's driving the disease, which we can do with these types of molecules, we call them siRNAs or guide RNAs. These guide RNAs can turn off a gene that's driving a disease. And there are several already approved. You mentioned inclisiran, but there are several others and many, many more that are in the pipeline that are saving patients' lives. And that's an incredible feeling. It's an incredible opportunity. But, I have to tell you, we need to pick up there's a lot of work that needs to be done. There are a lot of things, what I hate as a scientist and someone involved in trying to make a difference for these patients is that there's a gap between what's already possible and what's being done, right?
Right.
And that gap is what we need to close. We need to close that gap. We need to get the drugs in the pipeline and get them made. And the as probably everyone knows, it costs a lot to do a single clinical trial. It's so expensive, and yet there's so many that need to be done. So many new opportunities using RNA interference drugs to affect therapy. I can give you some more examples too, but I don't, if it comes up, I'm happy to talk more about the future because there's a lot coming.
Yeah, this is exactly where I'm heading to. And hearing you talk, so in my PhD, I worked a lot with drug delivery, and we spoke a lot with physicians here in Boston, in Brigham and Women's Hospital. And one of the main things that physicians would tell us is that the problem that they face is not necessarily the lack of new drugs, but delivery, delivery, delivery. And I recall some talks that I heard from you, Professor, that you also face a lot of challenges in the delivery of siRNAs based therapies as well. And so I would be happy to hear a little bit about how do you see the future of this type of therapies in light of the challenges they face, and also perhaps on a general level, how do you see the future of medicine, the healthcare that we will provide to patients in the near future?
Yeah, you definitely hit it exactly. A huge obstacle to this type of therapy has been delivery. And the all of the approved drugs target the liver. The liver, basically filters the blood. That's its job, right? I mean, it's that's where everything goes that goes into your system. It all goes to the liver right away or very quickly. So yeah, the drugs that worked at first were the drugs that were targeting activities or diseases of the liver where the gene that needed to be modulated was expressed or caused its pathological effect in the liver. So since then, however, there are breakthroughs, some of them from the RNA Therapeutics Institute that I co-founded at the University of Massachusetts from the laboratory of Anastasia Kurova, who is one of these RNA chemists who's been trying to crack open delivery outside the liver. That's been her focus for years. And her laboratory developed delivery to the brain that is quite good to, when you look at it's kind of gross, but after you deliver this to a mouse, you can cut their brain in half and you can look at where the guide RNAs went because you can put a little fluorescent tag on them. The molecules are getting into every single cell in the animal's brain. And when they enter the brain, they enter the cell by becoming transferred across the membrane in this process that is basically the normal way cells engulf their surrounding fluids. In the case of the brain, it's the cerebral spinal fluid that is where the guide RNA is delivered. So we have to deliver the guide RNA into the patient's cerebral spinal fluid through a spinal tap type injection. It's called intrathecal delivery. But once the molecule is delivered, it goes to every brain cell, including all of the support cells as well as all the neurons throughout the entire brain. That discovery led to the formation of a company called Atalanta Therapeutics, based in Boston, and co-founded by myself and Anastasia and another colleague at UMass, Neil Aronin. And Neil's lab had been working for many years on Huntington disease, which is a terrible neurological degenerative disease. The original siRNAs targeting Huntington were the basis for the formation of that effort. That company is about to enter the clinic with some pretty exciting therapies that we hope will have impacts on a variety of different neurological disorders, including Alzheimer's and Huntington, as well as ALS and some forms of epilepsy. So there are many, many opportunities to deliver that drug into the brain and make other companies as well have brain delivery working pretty well now too. So there's a lot of competition in this field, which is great. And we're hoping that these types of drugs for modulating uh, targets in the brain will soon be helping patients who, otherwise really have a grim outlook. If you're diagnosed with Alzheimer's disease or ALS, there's nothing for you, nothing that works very well. So we're very hopeful that these will be medications that actually stop the disease,
That's
halt the disease, stop the disease progression, allow the patient to continue with a fairly normal life and not have to face that horrifying prospect that would otherwise come with their diagnosis. So that's just one example, but there are many, many opportunities now to deliver these types of guide RNA molecules into tissues throughout the body. So there are really, really many new strategies, many new opportunities to make drugs, and so the future really is bright. There's some really good stuff coming.
that's absolutely fantastic, and yeah, we need a bright future for um, all the lag that exists still in the treatment of some diseases. So you mentioned the brain, the CNS. In my PhD, for instance, we worked with a blood-brain barrier disruption controlled via ultrasound to do delivery of therapies to brain cancer that are, as you probably know, brain cancer patients have a very grim survival and etc. So I have my fingers crossed. Um, Professor, I have a question here from Professor Lydia from Brazil. I will share now her clip. 1 second. Let me know if you can see her on screen.
I see her.
Awesome. So I'm going to play here.
Hello, I'm Lígia Pereira. I'm professor of human genetics at the University of São Paulo, and I head the Population Genomic Project DNA do Brazil. Dr. Melo, how do you see the study of gene regulation contributing to precision medicine when we consider the vast genetic diversity of non-European populations? And what steps do you think are most critical to ensure that discoveries in this field translate into equitable benefits across different genetic backgrounds? Thank you.
Very good question.
That's a great question. The To begin with, I should say that one really great thing in the favor of siRNAs in their more or less usual form, which is a, basically a molecular drug. It's not a complicated drug. It's not a cell therapy. It doesn't require, living cells or transfusions. It's not permanent. The drug goes away over time. So there's a lot of features about siRNA drugs that make them likely to have very broad impact. I already mentioned the long duration of effect. The cost of a dose is only a few dollars. So maybe 10, $20. When you start making these molecules at large scale, the costs come down. They're not very, very costly and they last a very long time. So the therapy is on par in terms of cost and access. Cost is a big barrier to access in many parts of the world, not to mention the genetic questions in the other in at the heart of this question. But the opportunity to essentially make drugs that are impactful, even where there's not a lot of money to pay for them, I think is really, really exciting. Now, the flip side of the genetic diversity question that was just raised is that for the most part, humans are fairly well uh, at the nucleotide level, quite similar within the regions that are targeted by all of the siRNA drugs that are currently on the market. There really aren't any drugs that have been made yet that won't work for every human being, as far as we know. Um, but there is a future where there will be some interesting new drugs. And I'll give you one example. There's a horrible disease called fibrodysplasia ossificans progressiva, which is always caused by a single nucleotide mutation. That mutation in a BMP receptor called ACVR1 causes these patients to make bone whenever they're bruised. So if they get a bruise in a muscle, these little mesenchymal stem cells that are present in their vasculature become activated by the inflammatory signal uh, to actually make bone. And these kids become ossified. Their entire skeleton, their entire body, they can't even breathe because their rib cage and the muscles around it literally turn into bone. But my colleagues at UMass, Julia Alterman and Jae-hyuk Shim, working together, decided to try to tackle that disease. You cannot knock down this receptor uh, with and have a viable outcome, right? The people need the function of this gene. So what they had to try to do was preserve the wild type uh, allele that differs by only a single nucleotide and silence only the mutant copy. So this is what we call a dominant, autosomal dominant mutation. These patients have one bad copy. And so the question was, could they make a drug that would shut down the bad copy without shutting down the good copy? And they did it. They've got a drug now that is ready meaning ready to go into the preclinical tox and development stage to show that it's safe in non-human primates and then move into humans. But this drug, if it works the way it does in the mouse model for the disease, which has it's a what we call a humanized, genetically humanized mouse. So it has the human ACVR1 receptor with the human mutation as a heterozygote. And that mouse becomes ossified whenever it's bruised. They can suppress this ossification phenotype. So if this works like it does in the mouse, this is a therapy that can be given to these patients, uh, hopefully prophylactically, so that they never become encased in bone in the first place. But later on, also when they're having surgeries, for example, to open up their jaw or their rib cage, it can be given to suppress new bone growth so that those surgeries won't have to be repeated month after month. Um, so that's an example, though, of where the future of these guide RNA molecules are going. We can now make guide RNAs, hopefully for many different targets that can discriminate a single nucleotide difference. And this does open the opportunity to make drugs for a host of different diseases that are present currently in humanity as a whole. And the question of how that development will be done equitably, I think is a really good one because as you know, many of these genetic syndromes affect, different populations around the world, because they're familial in origin quite often. So will they be developed or will they be way down on the list, of therapies that get developed? And I'm hopeful because of the relatively low cost that they will be developed. But, that's going to take a lot of work. It's kind of like if you remember the mRNA vaccine, which is relatively a much more expensive drug, but even so, manageable in terms of cost, that was not readily available to in many countries for quite a long time. So siRNA manufacturing has to be brought to scale to match the need. These guide RNAs are going to have major impacts on many human diseases. And you mentioned cancer earlier. Cancer is, uh, I think, going to be approachable. It's harder, as you know, cancer's tricky because it's constantly evolving. So, can siRNAs be used on cancer? Can we stay ahead of cancer? I think the answer to that is likely to be that we can use them, but they'll be part of a toolkit along with many other drugs. They will help trap the cancer cells in a blind alley, and then we'll get rid of them, right? We've got to throw everything we can at cancer, and I'm hopeful and eager to get going on that. There's really a lot more work to do, which you know I spend a lot, even though most of my effort is on this tiny little worm, I've been helping to raise money and get investment in this sector to try to get these drugs made and to get more of them to patients.
That's absolutely inspiring, and I think I hope you're right with all the possibilities that you just brought. I think especially in cancer, there are so many mutations that are already identified that confer tumor cells the ability, for instance, to repair after radiotherapy that perhaps we could tackle using siRNA approach. So yeah, I have again my fingers totally crossed for that as well. Uh, Professor, we have three more questions in about 10 minutes. Just a heads up here.
Okay, I'll try to be brief.
No, I don't want to. I mean, for us, it's been a completely blast. So it's just for your time. So I am a scientist, as I introduced myself to you before, and I also work part-time as a science communicator online. I like to bring science in a in a way that people can actually understand, right? And in this context, the question I would like to ask is that we have been facing, as scientists, difficult times with the denial of science in a global scale, especially on social media. So my question would be, do you believe scientists should speak out more? And if so, why is science communication important today?
That's a great question. I wish we had a whole hour for that one. We're really faced with a lot of headwinds right now, which is unfortunate because, you know, science is it, you know, it's a human enterprise. Science is nothing different really from what everyone does. Like whenever you learn something, you're basically using the scientific method. It's really simple, right? I mean, you gather all the data and you try to disprove your favorite model.
Yeah.
You're not gathering the data to try to prove you're right. You're gathering the data to try to prove you're wrong,
Yeah.
right? It's so simple. That's all it is. It's what everyone has ever done who ever discovered anything. I guarantee you that before people rode horses, everybody said you cannot ride a horse.
Yeah, that's true. That's true.
Somebody tried to disprove that, and that person took the
Became convenient.
scientific method. They
Yeah.
just tried. They tried it and it worked. Look, if you want to learn anything about anything, you got to be humble because you're going to be wrong, right? If you go in there loving your idea, trying to prove you're right, you will never be a good scientist. And we all know that, right? That's how science works. That's how discoveries are made. That's how good things happen. But in politics, it doesn't work that way. Ever admit you're wrong if you're a politician, you're done, right?
Yeah.
I don't know what it is about politics that makes it so different. I think a lot of, I think the United States was founded by a lot of a mixture of people, but some like Benjamin Franklin, who were clearly scientists at heart. So I feel like scientists probably need to be more involved in politics. I think it's really a steep hill for anyone to climb to be involved. I think being involved in our communities and just trying to explain, science so people can understand it because it does impact them, like vaccination, super important, really well established, and I guarantee that everyone at some point in the future will be wanting to be vaccinated because there will be something really nasty coming in the future. And unlike COVID, which didn't kill everybody and mostly killed old people or people you didn't know, if you had a really bad virus like a polio virus that cripples kids and or a smallpox that leaves horrible wounds all over your body, believe me, the lines would be around the block
That's true.
waiting for that vaccination. It will become fashionable again. It's just so unfortunate that lives will be lost, and unfortunately, opportunities will be lost because the politics of science. I don't know what happened. I don't know why in the United States, the current administration is turning away from science. Science is what made America great in the first place, right? I mean, their whole slogan is make us great again. Well, if you want to do that, you should double the amount of science funding, not cut it.
Mhm.
This is where the United States excels. We have been excelling in breaking open new ground in scientifically, and that's because of the investments that the federal government has made. And I'm just really sad that that's been lost sight of. And I think a lot of people in our government still get it. So maybe it's not over for United States science currently. Certainly, it's an opportunity for Brazil, provided that you can find the money and leaders with the vision to make an investment. Scientific discovery is an economic engine.
Yes.
Whoever is the best at it will have the strongest economy.
Yeah.
And medicine, healthcare costs are eating the budgets of every major country. If we can improve human health, we can affect incredible impacts at the economic level. So I do think politicians are going to be wanting this. They're going to
Yes.
and they'll want these drugs, because they're good ones. I didn't tell you about some of the really good ones that will hack our metabolism, let you eat whatever you want,
Oh my
uh,
God.
let you
Those are,
live to
those are the
be
good ones.
let you live to be 100, and still be active. Let's hope.
Yeah,
Um,
that's
it's our
absolutely important message, Professor. And yes, I wouldn't say it's only in America. I think this movement is everywhere, especially across internet. And I don't have a good answer as well as to why. And in this line of vaccines that you mentioned, RNA, that is your life work, I would like to invite Poliana. She's a journalist for one of the major outlets in Brazil. I'm going to share her clip. 1 second that I need permission, Tassi, to show. Yeah, perfect. So let me know if you can see her on screen. So I'm going to play.
Olá, meu nome é Poliana Casemiro, sou repórter de Ciência e Saúde no G1 da TV Globo. A minha pergunta é sobre se, na sua opinião, a desinformação sobre a questão da RNA pode acabar dificultando o financiamento de pesquisas e impedir o avanço dessa tecnologia. Se isso pode ser, pode se transformar no impedimento para o desenvolvimento de tecnologias que usem o RNA no futuro.
Yep. So
Can you paraphrase that question? I tried to read it.
So yes, so she asked if the misinformation around RNA technology could impair research funding. That's the first part of her question. Let me just play the second part to be sure I'm asking the full question.
Pesquisas impedir o avanço dessa tecnologia. Se isso pode ser, pode se transformar no impedimento para o desenvolvimento.
And she also asks if the lack of funding could become a barrier to the development of new technologies.
Absolutely. We're already seeing that happen. Sadly, in the United States, grants that use the term RNA, which is pretty much all of them, are getting extra scrutiny or even being denied funding. Sadly, I mean, it totally makes no sense. I mean, um, and so yes, anti-intellectualism in government is a huge barrier to progress. I mean, we've seen it over and over again through the history of human civilization, right? Cultures where science flourishes are cultures that thrive. So let's hope that will endure and that will return. I I do think it's likely that people will get it in the near future. It seems to me kind of incredible how, for example, popular and if you think about injectable drugs, vaccines are injectables. You inject an antigen. It's a very simple drug. It basically develops your immune response so that you'll recognize a pathogen when you get it. But then you have a therapy like the GLP-1 agonist, which is a injectable hormone therapy. That is so popular that they can't make enough of the drug. The
Yes.
company that made that drug, Novo Nordisk, became more valuable than the entire GMP of Denmark, where they're headquartered. I mean,
Boy.
uh, it doesn't make any sense. People want drugs that work, right? Vaccines work. People will want them in the future. I don't know what happened with this fear of things like autism that, if you inject 15 million people with anything, the next day, some of them are going to have some weird neurological symptoms, right? That's why we do the control of 15 million people getting the placebo, right? I mean, we've done this, we've been there, we've done this a million times, right? We're trying to disprove the idea that the vaccine is safe. And what we find is that they are safe. We
Fail.
can't disprove that idea. We've tried. Vaccines are good for humans. They help us. And, unfortunately, right now there's a backlash. Like I mentioned, my grandparents, they grew up with lots of really hard times. I mean, they saw polio. They saw they saw the devastation of polio. They saw the Great Flu that killed so many people during World War One or two. I can't remember which one it was, maybe both. But bottom line is people do appreciate medicines that work. They want medicines that work. And society needs medicines that work because they will improve the quality of life. And human health is so tightly tied to happiness. We need to be healthy to be happy. So I really feel, and I hope that this message will reach a broad audience in Brazil and across the world. I feel that scientists need to get out there and tell people that good stuff is coming. I mean, we have medicines that are planned based on siRNAs that will target genes that make us age faster or less well. That goes for the genes underlying dementia, right? Many people's lives are cut short by dementia, uh, Alzheimer's and related disorders. But another major cause of loss of quality of life and happiness during aging is loss of muscle and bone strength. We
Yep.
know a lot about the genetics of muscle and bone. We have siRNA therapies that are going to improve muscle and bone.
Wow, that's
They're-
amazing.
They are going to make, and these are based on human genetics, where we've learned from human genetics that people with mutations in genes like myostatin, for example, a major regulator of muscle wasting, this protein causes our muscles to waste if we are bedridden
Yep.
or starving. So the opportunity to make drugs that intervene that really dramatically improve the quality of human life, I think, is such an exciting prospect. Looks like you have another question queued up.
Yes. I'm gonna play now a question from the Ministry of Science and Technology of Brazil. A very nice question.
I can't hear the audio.
Yeah. Let me see if I can share myself here. 1 second. Let's correct that. 1 second, Professor. So I'm gonna try to share myself because I think the audio can come from my computer. Yep. So now I guess you will hear that.
Olá, doutor Melo. Sou Luciana Santos, ministro da Ciência, Tecnologia e Inovação do Brasil. E minha pergunta é: quais políticas ou programas públicos você considera que foram fundamentais para a sua formação científica e para a conquista do Prêmio Nobel?
Yes, more like a political program type of investment of science.
Yeah, well, we talked about this earlier. When I was a kid, we had the first humans walking on the moon back in 1969. I remember looking at the moon, that whole journey to the moon was inspired in part by the politics of Sputnik and the competition with the Soviet Union. We are a competitive species, humans. The United States invested a lot of money in science, in part because fear of the Soviet Union. I kind of expected our current administration would double down on science too, because obviously, the people who want to eat our lunch now are not just, the Russians, but the Chinese, India, Brazil. You guys all want a part of the pie that the United States has for years been, gobbling up, and that pie is scientific discovery.
Yep.
So for me, the stage was set by public policy at the federal level that made science a priority. When I finished college, I could go to graduate school at no cost. They paid me to go to graduate school. I got my PhD without having to do anything except the laboratory work that was part of my PhD requirement, right? So the opportunity came from wise federal government policy. And we need that very much because science is a costly enterprise. It used to be done only by the wealthy, sort of in their spare time.
Yep.
It was not publicly funded. It's done a little bit by the private sector, but around the world, most of the real science, most of the major scientific discoveries come out of laboratories that are government funded. So I benefited from that. I benefited from public education. We need wise policy, and science is a way of solving problems. It's the future uh, for um, for everything. It's where the good stuff comes from. So I hope that Brazil will double, triple, quadruple its investments in science. I'm 1/4 Portuguese on my Melo side
Yeah,
is
yeah.
from the Azores, so I'm sure I have many
Cousins.
cousins in Brazil,
Yeah,
and I feel like more than being an American, I feel like I'm a human being first, right? I mean, I'm an earthling. We all are. Let's work together. Let's break down these barriers that keep us apart. Let's solve the problems that are making people sick and shortening our lives and causing unhappiness and inequality. Let's try to solve these problems. That's what science is about, and we need a strong investment from the public sector and from just the public in general to make all that happen. Let's do it together.
Yes, I remember one of your talks, you mentioned that we scientists, we are citizens of the world. We're not like Brazilian, American. We are scientists, human beings, right? We care about the world first, then everything else. And I really appreciate that statement. And also, I would like to mention another statement that I heard that I think it totally resonates to the message you just gave. It is a famous sentence. I forgot the name of the person who said that, but if you think science is expensive, try the disease, right? You probably heard that one. So yeah, we need totally to invest in science. And Professor, the last question, I apologize, we are a bit over time, but this is a very important question from students, high schoolers, that they want to ask you a very important message. I'm going to ask Tassi just to let me share the video. Yes, perfect. So I'm sharing right now.
Hi, Craig. I'm Sophie.
And I am Pedro.
We're from Montes Claros, Minas Gerais, and our question is:
What are the biggest challenges that you faced during your career in science?
and what are some advices that you would give to young people like us who want to pursue the same career in medicine? See you Thursday.
Bye bye.
Yes. So
Yeah.
Sophia and Pedro asking from Minas Gerais. You probably heard it's
Yeah. Thanks for that question. You know, I have to say I've been incredibly lucky as a scientist. I think one of the things that you know, if I tried, I often get asked for advice, so I always try to do my best, right? You know, I do feel like you just have to be lucky at some point, but I think you also kind of make your luck, right? I mean, if you believe in yourself, if you believe you can do it, then you will try hard, right? You I always tell people don't believe anything, but believe in yourself, right? Don't believe anything you read. If somebody says an experiment won't work, do it anyway.
Yeah.
Right? That's basically what I'm trying to say. Like if you have, you know, like if, and not everybody gets the opportunities I got. I feel again incredibly fortunate because, you know, I got these opportunities and I if I hadn't gotten them, I would have had to find another job like my grandfather who didn't get to finish high school, right? I mean, my grandmother, both my grandfathers, they just had to go to work and then they had to make the best of their lives. If you get the chance and you're in school and you like science, go for it. Believe in yourself. Believe that you can make a difference. Try to understand things and ask questions. You know, try to get the answer. Try to, you know, break it down into pieces and solve it. You know, get your foot in the door. I didn't get to tell you about the really cool stuff we're working on now, but there's so much still to discover. There's so many incredibly cool possible future scenarios for understanding the biology of how organisms handle information. We are only scratching the surface of understanding that. There are going to be huge breakthroughs in the biological sciences in our lifetimes, in their lifetimes for sure. I hope in my lifetime too. But yeah, become a scientist, but believe in yourself, work hard. As long as you've got the opportunity, just do your best and don't give up if you can. If you can just find a way, science is really, really fun. Go for it. That's what I would say.
That's absolutely inspiring, and I think that will for sure inspire a lot of young people, but not only young people, myself, that I'm not that young anymore, Professor, and other folks. We really will be happy, and we dream that a Brazilian will eventually also be laureated with the Nobel Prize. We have so many good scientists, very high, knowledgeable.
I have only one request.
Yes.
Whoever you are, when you win the prize, please invite me to the ceremony. I want to come. It's an absolute blast. We will dance the night away together and celebrate your discovery. I look forward to that day as well.
That'll be amazing. Professor Melo, I am so fortunate as well to be here talking to you. It has always been my dream to just chat, with someone like you. And I really, really, really appreciate your time today. You can be positive that your words will resonate to several dozens of thousands or hundreds of thousands of people across Brazil and the world, especially because the language we're speaking is in English, a universal language. So once again, I would like to extend our utmost thank you. And if you have any other comments, feel free to just say now or thank you again.
Yeah, my pleasure. And I look forward to maybe meeting many of you in person at some future event in Brazil.
Absolutely. We will coordinate that for sure.
Good.
Thank you very much, everyone. It has been a tremendous event. So we will keep this recording and we will upload to YouTube for those who don't speak English. There will be a translation there, but I hope everyone could learn a lot as I did, as everyone else did. And thank you for your attendance.
Thank you,
Professor,
everyone.
bye-bye. I will email you with the second link now, okay?
Excellent. Take care. Bye, everyone.
Bye-bye.